What walks on four legs in the morning, on two legs in the afternoon, and on three legs in the evening? The answer is humans. This is because aging has been taken as a universal truth. But is aging truly universal? Can it never be reversed? If the answer is no, then we humans don't accept that. For centuries, humans have been in search for a cure for aging. Various mythical quests have been made, but today's modern day research is more focused on the biology. Now, by observing the DNA modification and various gene expression throughout the ages, we try to understand aging. We observe various biomolecules and how they change through aging. This has led to invention of various aging clocks. Most notable are transcriptomic clocks and methylation clock. Now, transcriptomic clocks rely on the gene expression and how they change through aging, but we focus on the methylation clock. Now, what is methylation clock? DNA methylation is the addition of methyl group to specific sites in the DNA. By addition of these methyl groups to the DNA in those specific sites, CpG sites, they regulate our gene expression. And this in turn affects our aging. Now, the specific sites where methylation occurs are called CpG sites, cytosine and guanine. When these cytosine and guanine occur in large number in a large sequence, they are known as CpG island, and they are very crucial, as we will see. The first ever clock was made on a saliva for methylation. By observing those changes, they were able to predict how epigenetic age changes with our chronological ages. The next big step was Horvath's clock. Now, Horvath made another step by introducing a pan-tissue epigenetic clock, which was more opposed to the tissue approach, where he showed a remarkable progress with the chronological aging and epigenetic aging. But this was a very simple model. This was based on linear regression. Now, we're not going to say that linear regression is a bad choice, but it was a very simple model. Then down the line, scientists made approaches to deeper models by using deep learning models like neural networks. Ultimate was one of them. And the most notable thing about them was they used SHAP to interpret how this model worked. Now we have to understand another big concept, that is hypermethylation and hypomethylation. That is just how methylation changes. Hypermethylation is increase of methylation, and hypomethylation is a decrease of methylation with age. Now this also affects our gene expression and aging. So this is an important concept that our model needs to know. Next, we also need to understand co-methylation. Co-methylation means when two CpG sites affect each other's level of methylation based on their methylation. This important concept needs to be incorporated into our prediction for a more biologically relevant prediction. But how do we actually do this? This is a research gap that our research has tried to fill. Another thing that we need to point out is there are more biologically relevant data, like is it a CpG island? If so, what is the length of that island? What is the next base pair? So these information are not included in other models. Our model is the first to have incorporated all of this into its own representation of input. As a result, we would get a better interpretation. But how do we actually do this? For this, what if we have this tabular data and we convert this tabular data into a graph? By converting this tabular data into graph, where all the CpG site information, biologically relevant information are node and the relationship as edges. The node attributes will have a methylation. Is it a CpG island? What is its length? What is the next base pair? what is the starting position of that island and ending position, what is exact coordinates of them. All of them will be included. And the edges will include the co-methylation and the relationship, like are these two CpG sites in the same gene, are they in the same chromosome, will be included in the edges. Now, after converting this tabular data into a graph, we need to filter some of the edges out for computational ease. For this, we have employed 3 filters. One filter is a universal filter, and the rest 2 are chromosome specific and distance specific. Anything less than this filter are filtered out. Now, the reason for chromosome specific filter is because closer CpG sites have more influence of co-methylation. This is why we need a lower filter of chromosome specific, which allows us to include edges with lower co-methylation value but which are closer together for more better interpretation. Now, if we come to what our model actually looks like, the architecture, it is first, after we've converted our tabular data into a graph representation, we feed that to a graph neural network in a stochastic way. After that, the graph neural network gives us an embedding. That embedding is given to a fully connected layer. That fully connected layer gives us the final age prediction. This gives us an interpretation, a model that takes into account all the structural and positional information to make a good prediction, and the interpretation of this prediction will have a more biologically relevant interpretation. Now for the results. As you can see, our model is quite competitive. After doing a 5-fold cross-validation, we've compared it with the model like Ultimage, and you can see that the performance is quite competitive, even performing slightly better in the test case. Another observation is that as the models are progressing with age, we can see that the error is increasing. This definitely shows us that as we age, our epigenetic noise increases, which makes it harder for the model to give a better accurate prediction. As a result, the epigenetic age is more different as we age. Now for the most important part, how do we actually interpret this model? We use GNN explainer to do this. It gives us 3 types of importance: the node attribute importance, the node importance, that is the CpG site importance, and the edge importance, that is the relationships importance. After getting the attribute importance for each of the sample, we take an average of them. From the given picture, we can see that after we have averaged them, the mean is given. The most important attribute is methylation, which was pretty obvious. Next, we can see that the next base pair is C, is very crucial for the age prediction. After that, we can see that is it a CpG island, what is its starting position, what is the ending position. That is CpG island related information is very important. And the next base pair is C, is also very important compared to other base pair. Now you can see that if we plot the importance of each attributes with their age, we get a temporal analysis of the importance of those attributes. We can see that 2 attributes, distance from transcription start site, and the other one is the exact location of that CpG site, that is map info, have increased their importance up to age 20. After that, we can see that there is a sharp decline for these specific 2 attributes. On the other hand, the other attributes such as length of the CpG site, is it a CpG site, the next base pair is C, A, or T, all this information have an increase. They actually first go down up to 20, then they have an increase. This gives us an understanding of how various biological information affect our aging and how they affect our aging in different ages. Now, GNN explainer also gives us node importance and edge importance. We filter out all the zero important nodes and all the edges whose importance are less than 0.1. After filtering out and removing those nodes and edges, we get a few connected CPG sites whose importance are very influencing to our age. These subnetworks of CPG sites are collectively what we call methylation regulated network. The reason behind this is that these CPG sites influence each other's methylation level, and that in turn regulates the gene expression. So this complicated and complex network has been very intuitively been visualized through this graph, and you can see this is one single subnetwork of our methylation regulated network. Now, depending on the graph, there might be similar kind of subnetworks in that particular methylation regulated network. And based on the color, you can understand that red color nodes are hypomethylated and blue color nodes are hypermethylated. This gives us a better understanding, and the green color circle nodes have their importance increasing with age, and yellow color circle nodes have their importance decreasing through age. There are also other annotations in those graphs which help the researcher understand more, like what is the gene, what is the CPG site name, what is its importance, how much hypermethylated or hypomethylated it gets with age, then what is its distance from transcription start site. Also in the edges you can see annotations like what is the co-methylation between those sites and what is the importance of that particular relationship. So this gives a researcher more broader understanding. Now, for pathway analysis that most models do, our model gives a more deeper understanding that we take that these connected networks are more similar. So we take the connected networks which are hypermethylating through age, and we do pathway analysis, and we also do the same for ones that are being hypermethylated. So for both cases we do pathway analysis separately. Now, after doing pathway analysis for the hypermethylation CPG sites gene, we get that cardiac muscle and strident muscle pathways undergo hypermethylation. That is, the gene expression tend to decrease with age. This is a big understanding of our knowledge about aging. And after doing pathway analysis for hypermethylation, we understand that ovarian stridorogenesis pathway undergo hypermethylation with age. That is, their gene expression increases as they age. We also did a temporal analysis of the importance of the CPG site as the age progresses. We used a linear regression with the importance and the age to predict the slope of those CPG site importance. As you can see, we have given the top 10 downward trending CPG site importance. And here is the CPG sites whose importance experience upward trend as we age. Now, a few CPG sites do not experience such kind of fluctuation as we age. Their importance remain constant throughout age. This is some set of CPG sites whose importance remain constant even as we age. This gives a more understanding that different CPG sites experience different kind of importance at different ages. Now, a final few words is that now is the time for us to come together. We can see that there is no data set where DNA methylation, gene expression, and DNA mutation is present. We need to come together and create a unified data set that will have all this information of a single person. This will help us understand aging. And with this understanding, we can accomplish the impossible. Cure aging. This is a big leap for humanity, and we will be part of history.